The overall goal of this procedure is to analyze intracellular gene expression directly in living cells via the application of gene-specific fluorescent nanoparticles. This is accomplished by first defining the appropriate target sequences in the gene of interest. The second step is to manufacture nanoparticles with specific capture and reporter strands. Next, the nanoparticles are added directly to the culture medium of the living cells. The final step is to incubate the cells with the nanoparticles overnight at 37 degrees Celsius and 5% CO2 in a humidified atmosphere. Ultimately, fluorescence microscopy is used to visualize gene-specific fluorescence in living target cells, while flow cytometry analysis is used to quantify the number of fluorescent cells.
The main advantage of this technique over existing methods such as immunohistochemistry, molecular beacons, or PCR analysis is that gene expression can be observed visually in live cells without any manipulation of the cell culture. Demonstrating the procedure will be Martina Dreßen, a biologist and PhD student from our laboratory, Lynette Henkel, for the flow cytometry.
After determining the target sequences, order lyophilized gold nanoparticles from commercially available sources. These particles are composed of a central gold particle covalently linked to a capture strand, which is bound to a reporter strand.